everyone, and welcome to lab number 6, which focuses on transformation, which I know it sounds like something very mystical and fairy tale like, but in reality, transformation is one of the main forms of gene transfer possible with microbial cells. And so, for instance, when we say gene transfer, we mean getting new DNA into these little valuable bacteria to help us out, okay? So we're going to go through what exactly gene transfer means, and then we'll go through the experiment of transformation. Now, the good news is you have finally made it past all of the annoying calculations and formulas, and now we're hitting the more conceptual aspects of genetics lab. The first thing that I want to mention is in microbiology, we kind of teach you that bacteria aren't always bad. There are a lot of good, helpful ones in your normal flora, and bacteria can be quite valuable and helpful in the realm of research, in the realm of product development. So, the food industry, the medical industry, all over. Some of the ways that they are valuable to us is their ability for bacterial gene transfer, meaning the idea that you could get new DNA, new sequences, new genes put into a bacteria, and you could then have them replicate it, make lots of copies, really play around with the genetic code and get bacteria to be nice little factories for you, basically. Now, there are 3 forms of gene transfer that I want you to know. You have to know the difference between each of them. The first one is transformation. And in transformation, the way we define that is the uptake of naked DNA by bacteria. Okay, so what that means is basically you have a bacteria that needs to be made competent. So meaning that it's able to be, ready to take in DNA. And then you have regular old naked DNA, meaning just strands of DNA around that cell, not in any kind of structure or cell, just free-floating DNA, and you get the bacteria to take that DNA in. The other form of gene transfer that you see on this slide that sounds a little similar is transduction. And the difference with transduction is that the bacteria is still taking in new DNA, but the way it's doing it is by virus vector, meaning a virus injects new DNA into the bacteria. The third form of gene transfer that we have here is conjugation, and my students like to remember it as conjugal visits. Okay, if you don't know what that is, I will not explain it, but for those of you who do know what that is, it's a little trick that can help you remember that conjugation is basically a fancy way of saying bacteria sex, because as you can see in this third picture, in conjugation, one bacteria cell is getting DNA from another, and the way they transfer it or they get that DNA is through the formation of a sex pilus, basically a little narrow tunnel for DNA to transfer from the donor to the recipient. Okay, so when you look at these 3 forms of gene transfer, ask yourself, how is the bacteria getting the new DNA? In transformation, the bacteria is getting pure DNA from the surrounding area. In transduction, the bacteria is getting new DNA from a virus. And lastly, in conjugation, the bacteria is getting new DNA from another bacteria cell. In case you missed any of that, you can pause on this recap and jot down some notes. Okay, so now that you're all experts on the 3 possible types of gene transfer when we talk about bacteria, Now we're going to focus on our experiment today, which is transformation. And now a lot of you have probably done transformation before in previous classes. You know, transformation is one of those very versatile experiments that no matter which part of the sciences you go into, you end up using it tons of times. And that's why we cover it in, you know, courses like Bio 171 or 181. We cover it in genetics lab. We do it again if you take molecular, such as Bio 327, 325. If you take cell biology, we do it there as well. We basically have you do transformation quite a few times because that's one of the experiments that you really have to get a good grasp on, you know, what it means, why we do it, how to do it, because you end up using it so much no matter which field in science you go into. Now, when we say transformation, the textbook kind of definition is the uptake of exogenous DNA, usually through vector or plasmid DNA, into cells, specifically bacteria. Now, I know that sounds all fancy and scientific, but basically what it means, exogenous. When you hear exo, like exoskeleton, you know that means on the outside, right? So that means outside. So the uptake of exogenous DNA tells you that you are getting these bacteria to take in DNA that's outside of their cell. Okay, so it's naked, just pure DNA strands in the outer environment of that cell. Okay, and when I say environment, I don't mean trees and plants like you think of our environment. I simply mean right near the outside of that cell. Okay? Now, in order to do this, cells need to be competent, okay? Unlike a lot of students I know. But anyway, competent, what that means in terms of the bacterial cell, we're going to draw this out in a picture later in these slides. But for now, just know that competent means getting the bacteria ready to take in outside DNA, okay? Exogenous DNA. Now, basically what that means is normally they wouldn't want to take in that DNA because of electrical charge issues that we're going to go into in more detail. But by making them competent, you get them ready to take in the new genes. Now you may ask yourself, okay, we do transformation so much in the sciences. Why? What's the purpose? Well, transformation's purpose is to allow us to get new genes, okay, new foreign genes into bacterial cells. Once that new DNA is in those bacteria, what will the bacteria do for you? They're like little factories. They're going to make tons and tons of copies of that new gene, of those new genes if there's multiple of them. And they will then ultimately produce the proteins that those genes code for. So now you have these nice little factories capable of making tons of whatever gene, whatever protein, meaning valuable enzymes, you want lots of copies of. Okay, so for instance, in the medical field, this was one of the first ways that they got a lot of insulin produced. Okay, so it's a very valuable technique. Now, in order to do this technique, you notice that the definition I gave on the previous slide says that usually that foreign or exogenous new DNA is in the form of a vector or plasmid, which is simply an extra little ring meaning circle, of DNA. Okay, we call it a cloning vector. Now, in order for your transformation to go well, and in order to get, you know, those new genes, the new DNA into those bacteria and efficiently, effectively reproduce it, you want to pick a cloning vector, sometimes or a lot of times called a plasmid, that has 4 main qualities to it. Okay? First of all, the cloning vector or the plasmid should be fairly small DNA. And you want to ask yourself, well, why is that? Well, think about it. What do you want to happen in a transformation? You want the bacteria to take in that little round plasmid DNA. If it's large, good luck getting that giant piece of DNA into those teeny light teeny tiny little bacterial cells into their teeny tiny little pores on the outside of their outer cell walls or membranes. Good luck with that, and it's not going to go very efficiently once it's inside of that little cell. So you want the vector to be fairly small DNA. Why? To help it get into the bacteria. Okay, help it get into the bacteria. Secondly, it should have an origin of replication. Why is that? Well, you want the bacteria to do what? Make a whole lot of copies of that DNA for you. Okay, so by having an origin of replication, it allows the bacteria to make lots of copies for you. Okay, it maintains that plasmid, those new genes, in the bacterial population, which is really valuable to you, because what good would it be if you got one piece of DNA into one bacteria and that's it, that's the end, it couldn't replicate it. It'd be useless to you, waste of money, waste of time, okay? So origin of replication is important. It should also have at least one selectable marker. Okay, we're going to go through the selectable markers we have in our experiment, but an example of a selectable marker might be antibiotic resistance, or genes for color changes, or even fluorescence. Okay, so examples of selectable markers are, we're going to go through the specific names like beta-lactamase and beta-galactosidase later, but examples would be genes that code for color changes, fluorescence, or antibiotic resistance. The reason why you need these selectable markers is they allow us to detect which bacterial cells successfully took in the new DNA. Okay, so they are for detection of a successful transformation. The last thing that you want in your cloning vector is that it should have at least one unique endonuclease recognition site. We call those restriction sites. We're going to talk more about that in the lesson that we have on restriction enzymes. But for now, think of it as multiple cloning sites, or MCS. The reason that you want these multiple cloning sites or these restriction sites is it allows you to cut open that plasmid or that vector and manipulate the DNA. You can then insert whatever genes you want into that ring of DNA and pop it into the bacterial cells. Okay, so why should you have a restriction site on the cloning vector? It allows you to manipulate and add new genes to that vector. Very valuable, very powerful. Now I keep saying the term plasmid or vector, and I want you to know, well, what exactly does that mean? Well, the official, textbook kind of definition that I usually give students is circular, small, extrachromosomal genetic elements that are not essential for bacteria to survive, but often confer advantageous traits. So basically what that means is, in bacteria, you have the big old chromosomal DNA all clumped up in its nucleoid-looking structure. Remember, no nucleus because it's a prokaryote, but instead it's more of a nucleoid-looking clump of DNA. And that's all necessary for life functions, everyday survival of the bacteria. In addition to that chromosomal DNA, bacteria can have extra little rings of DNA, so little rings containing a various amount of extra genes. Now, they are extra chromosomal, which means that they're beyond or separate from that big old chromosomal life-functioning DNA that every bacteria has. That means that, not every bacteria will have a plasmid. The ones that do have plasmids will not all have the same ones. They're extra chromosomal. Now, because they're not part of the regular chromosome, they're not essential for everyday survival, meaning normal everyday life functions. But they often do confer advantageous traits. And with that, I'm going to jump to the last question that I wrote on the slide. what do we mean by advantageous traits? What advantage can a plasmid occur? Well, the big one is antibiotic resistance, which is shown here. Okay, so plasmids can contain antibiotic resistance genes. And if you take microbiology, we go through all of the various specific ones. Now, with the antibiotic resistance genes, well, think about it. That's not necessary for everyday, normal, essential functioning. But in the event that that bacteria suddenly encounters antibiotics, well, now it has an advantage for survival. Okay? Only the ones that have that particular plasmid will survive under those conditions. Okay, so the big advantage that plasmids can confer is antibiotic resistance. Now, the other aspect of plasmid definitions that we usually mention is that plasmids are self-autonomous replicons. What that means is they can replicate by themselves. How, might you ask? Well, you already know the answer to that thanks to the previous slide. They're able to replicate by themselves because they have the origin of replication that we mentioned in the previous slide. Okay, and you'll go more into that in detail in the lecture part of this course. Now, basically, self-autonomous replicons means that the plasmids can replicate a whole bunch of times by themselves in the bacterial cell rather than having to wait until that bacteria doubles its normal chromosomal DNA and then splits. Okay, there are some plasmids that will only replicate in step with the chromosomal DNA, but in general, a lot of them are self-autonomous, meaning they can replicate separately from the chromosomal DNA. Now, in our particular transformation experiment that we do in the genetics lab with the students, we use the vector called pBlue. So if I ever ask you which plasmid, which vector you use in your experiment, it is pBlue. And which selectable markers does it have? Well, it has two very important genes for our experiment. It has a gene for beta-lactamase and a gene for beta-galactosidase. Now, what that gene for beta-lactamase allows it to do is ultimately that gene codes for the RNA that codes for the protein to make the enzyme beta-lactamase, which cuts what's called the beta-lactam ring in the chemical structure of certain antibiotics such as penicillin and ampicillin. Okay, so as you can see in this figure, beta-lactamase cuts the beta-lactam ring of certain antibiotics so that now that antibiotic is in an inactive form, okay? Because normally antibiotics like penicillin and ampicillin, for instance, they use that beta-lactam ring in order to bind to the enzymes that help the bacteria build their cell walls. And so if you then cut that beta-lactam ring open so that it's now an open chemical structure, that antibiotic can no longer bind to the bacteria's enzymes and it can no longer kill that bacteria. So you have now, with this gene, made that bacteria antibiotic resistant. And we can visualize that in the results by growing the transformation samples on plates that contain or lack ampicillin. Okay, and so we'll go over that in more detail when we go over expected results. But basically, if a bacteria takes in pBlue, it should now be able to produce beta-lactamase. And notice I say the pBlue contains the gene for beta-lactamase because pBlue is DNA. The vector, the plasmid, is DNA. So it cannot cut antibiotics. It is the gene that ultimately makes or codes for the protein beta-lactamase that then inactivates the antibiotics. Please circle star highlight that fact because it comes back when we go over expected results and when you have to analyze parts of the protocol. Now, like I said, we visualize that based on plates with ampicillin or without ampicillin. If bacteria took in the P blue vector, it should now be able to produce beta-lactamase, which means that if you grow it on a plate with ampicillin present, we usually write it as AMP, AMP. If the plate has ampicillin, these newly transformed bacteria can now cut that ampicillin and survive on that plate when they would have been killed previously. In addition to that, the P blue vector also has a gene for beta-galactosidase. That is responsible for color detection. So what beta-galactosidase does, ultimately, when you talk about genetics, you see beta-galactosidase as being part of lactose metabolism. Okay, now beta-galactosidase is able to cut disaccharide sugars into their monosaccharide parts. Now, what scientists did was they created a fake or a special kind of sugar molecule called X-gal. So it's the letter X, G-A-L. And X-gal, when that is cut in half by beta-galactosidase, one of those halves has a blue color to it, okay? So beta-galactosidase allows us to visualize which bacteria took in P-blue and are successfully, working with this DNA based on which bacteria turn blue in the presence of X-gal. Please, circle star, highlight the fact that they will only turn blue if they have P blue and, circle star, underline, and if X-gal is present on the plate. So we label those plates as LB, which is the regular nutrients, and we put LB AMP because they have ampicillin in them, and X-gal. If the plate does not have X-gal in it, okay, that means that the bacteria, it could have all the P blue it wants in its cells. It will not make anything blue if X-gal is not present, because the blue color comes from beta-galactosidase cutting X-gal in half. Okay, please make sure you're clear on both of these selectable markers, their names, what they do, and how we visualize them. And please be very clear on the fact that if the bacteria does not have P blue, then it will not be able to survive in the presence of ampicillin, and it will not be able to produce blue cells in the presence of X-gal. Please note that if it has P blue but there's no X-gal present, you're not going to see the blue color. Okay? If you have any questions, you're uncertain of these markers, please contact me and remind. So now that you know the purpose of what you're doing in this experiment, and now that you know which plasmid you would be using, now it's important to go through the key steps of any transformation, because when you do a transformation, each protocol might be slightly different in terms of the exact number of seconds that you do something or the exact volumes that you use. But the main steps are always the same. And the first big one is something that we've kind of mentioned already, but now we're going to go into more detail, is the fact that the bacterial cells need to be made competent. And again, what competent means is they have to be ready and willing to take in that new plasmid DNA. Now, in order to do that in our experiment, what we do is we soak the cells in ice-cold calcium chloride. And when that ice-cold calcium chloride is, soaking with the cells, the calcium chloride dissociates into calcium and chloride ions. Now, what's very important with that is that those calcium ions are positive. Now that brings me to the next questions that I ask on this slide, which is, when you think about this process, what charge does DNA have? Meaning, what charge does that plasmid have Well, DNA, because of the phosphate groups, is highly negative. Okay, write that down, circle, star, highlight it. DNA is highly negative. The bacterial cell, due to the lipopolysaccharides in its cell membrane, the cell membrane is also highly negative. Now, what do you know about two negatives? They repel each other. Only opposites attract. So having negative DNA in a negative bacterial cell, when you look at it, the bacterial cell is not going to want to take in that negative DNA. The DNA will want to be as far away from that bacterial cell as possible, not go toward it and enter it. So that's why calcium chloride or other competency steps are so important. Because what happens is when you add the calcium chloride and it dissociates into calcium ions, you now have highly positive calcium that coats the negative bacterial cell, so that in the next step when you add the plasmid DNA, the DNA now thinks that bacterial cell is highly positive because it's wearing, think of it as wearing a coat of positive charged calcium ions. And as you're going to see when I draw this out on the next slide, the negative DNA is now drawn to or attracted to the positively charged cell. And again, it's not that the cell itself is positively charged, it's that it's currently coated with the positive calcium ions, which serve as what we call an ionic bridge. And on the next slide, I want you to circle, star, highlight that. Okay, so pay close attention to the picture on the next slide and make sure to draw it out just like I do to help visualize it, because this concept of competency, calcium ions, negative DNA, negative bacteria, this always comes back in the post labs, in the quizzes, in the final, in various other places. And like I said, this comes back in later semesters when you take other science labs. So now, in a gene transfer experiment like transformation, where you have DNA coming from the environment around the cell where it's not in any other structure, such as a virus or another bacteria, you have to make sure that you make the cells competent. And competent means ready to take in new DNA, okay? Meaning you have to make the cells the proper charge. So I'm going to draw out what we mean by this. When you have a bacterial cell, okay, so bacteria, you have that outer membrane of the bacteria, and that outer membrane is highly negative because there's a lot of lipopolysaccharides. Okay, so the bacterial cell is highly negative. Now you want that bacteria to take in DNA from the environment, and what do you know about DNA? What charge does DNA have? Well, because of the phosphate backgrounds, DNA is highly negative. Okay, so you have bacteria that's negative, and you have DNA that's negative. What's the problem with this? You want the bacteria to take in that DNA, but right now, what do you know about same charges? They repel each other when the charge is the same. So right now, that DNA doesn't want to go anywhere near that bacteria cell. So how do you get that DNA to become attracted to the bacteria? You make the bacteria competent by coating it in positive charges. And one of the ways we usually do that is by adding calcium chloride. So can add calcium chloride, and what that does is the calcium, when you put that calcium into the transformation solution, the calcium and the chloride separate from each other, and calcium is highly positively charged. So those calcium plus 2 ions, they coat the bacterial cell. Okay, that's all these plus charges that you see. Those are calcium ions coating the bacteria cell. Okay, so calcium coats bacteria in positive charges. And so we call that creating an ion bridge. Okay? Because these plus charges are a bridge connecting the negative DNA with the negative cell. Okay? So the calcium ions are a positive bridge. Okay? So we call that giving the cell chemical competency. All right? So with transformation, anytime you hear that term, please remember that you must first make the bacteria competent. Please understand what that means. So competent is making the cell ready to take in new DNA by affecting the charge of the cell. Okay? Make sure you remember that the bacterial membrane is negatively charged, the DNA is negatively charged, so they normally repel each other. You add calcium chloride, you add positive charges to create an ionic bridge to kind of bridge the two together, to make the DNA now highly attracted to that bacteria because it sees the bacteria now as positive. And as you know, opposites attract. So the negative DNA is now attracted to the positive bacteria. If you have any questions on this, please contact me in the Remind app. So now that you have a strong understanding of what competent cells means and how we make them competent, kind of to recap, the first thing you would do in a transformation is you would mix bacterial cells that you scoop off of a plate with some liquid calcium chloride, let them, sit for a bit on ice. And mixing those cells with the calcium chloride, you now have bacteria coated in positive charges. You would then mix some of those competent cells with plasmid DNA. In our case, what's the plasmid? It's pea blue. So you would mix the competent cells with the plasmid DNA because now you want that plasmid DNA to coat or stick to the outside of those competent cells. Again, sticking to the outside of the cells because those cells are currently coated in positively charged calcium. What you then would do is you would take that sample and you would put it into a hot water bath. I know, sounds relaxing. You would put it in the hot water bath as a heat shock step. And you only do that for, 45 to 50 seconds. Each protocol varies. Some of them are up to 90 seconds. Either way, it's not very long. It's about a minute that you keep it in 42 degrees Celsius. The reason you do the heat shock step is because that heat opens up the pores, those pores meaning those nice holes on the outside of the bacterial membranes, okay? So it opens up the pores of that bacterial cell, and since you currently have that bacterial cell coated with the plasmid DNA sticking there, this helps force the plasmid DNA into the bacterial cells. Okay, so the heat shock step is to open up the pores of the bacterial cell so that you force the DNA, the plasmid DNA, into the bacteria in this step. You then quickly return those bacterial cells to ice. Now think about it. Why would you want to do that? Well, you don't want those pores, those holes, staying open forever, right? So the point of the ice, now that you hopefully have the plasmid DNA in the bacteria because of the heat shock step, the point of the ice is to then close the pores. And when I'm saying pores, that's P-O-R-E-S, okay, pores. So you want to first do heat shock to force the DNA into the bacteria, you then return it to ice quickly to close up the pores and keep the DNA in the bacterial cell. After that, at this point, you hope that you now have the plasmid DNA nice and safely in the bacteria. You then incubate those cells in nutrient-rich media, which we call LB. So let me grab the pen and write nutrient-rich media. What we use is LB. That stands for Luria bertani. Okay, you don't have to memorize that, but just so you know, LB stands for Luria bertani. It's a type of broth or agar plate, and it basically has all of the nutrients and important things for bacteria to survive and replicate. Now, the reason why you incubate the cells at this point in nice nutrient-rich media is not simply a recovery phase. Think about it. The plasmid that you just put into those cells, what is the plasmid? It's DNA, right? Well, will that DNA as DNA protect that plasmid from the antibiotics you're going to use later in the later final steps? No, the DNA cannot cut those antibiotics. What cuts them is the enzyme or the protein made from that DNA. So the big significance of this step, the nutrient-rich media incubation, is to allow the cell, the bacteria, to replicate and transcribe and translate the plasmid DNA. Okay? The big significance is that the cell needs time to make the selective proteins, okay? The selective proteins of that plasmid. Specifically, in our case, it needs time to make the beta-lactamase and the beta-galactosidase, okay? So if you do not incubate in nutrient-rich media, then in the later steps, when you have antibiotic on some of the plates, you will kill the bacteria whether it has the plasmid or not. Okay, so please circle star highlight the nutrient-rich media incubation step, and remember that it is not simply recovery. It is so that the bacteria will replicate, transcribe, and translate that plasmid to make the protective proteins. Okay? Once it has had time to do that, you can then plate your samples on your various selective media and plates, and then incubate them to give them time to grow and produce visible colonies. Now, in order to confirm that our particular plasmid, which again, remember, our plasmid is P blue, okay? Our plasmid is P blue. Which plates will we use to confirm that plasmid? The main plate that will confirm success is the LB AMP XGAL plate, okay? LB is the nutrient-rich media that all bacteria love, has everything they need to grow, and any bacteria can survive on. Ampicillin is our selective aspect to test for beta-lactamase presence, and XGAL is our selective item to help detect whether the plasmid is in the bacteria because of beta-galactosidase. Okay, so we're going to go through the expected results, and we'll go through these plates again, but basically keep in mind when you see this plate, AMP and XGAL, that is how you truly confirm the presence of P blue and a successful transformation, because it tests for both selective markers, okay? The selective markers are beta-lactamase, the gene for beta-lactamase, and the gene for beta-galactosidase, okay? So by having AMP and XGAL, you test for both. Now, before we go through the expected results and the protocol, the last thing I want to mention here in the background information is that what you need to do whenever you do any experiment is you need to have a control group, okay? So you need to have a control group. You'll have a control group and you'll have control plates, which we'll talk about in the expected results. So our control group in our experiment will be minus P blue, meaning samples in which you do all of the steps of the transformation, but you do not add plasmid DNA. So you just have the bacteria, you add the calcium chloride to make them competent, you do the heat shock and the ice and the nutrient-rich media steps, and you plate them on selective media, but they never got exposed to plasmid DNA. So they are original bacteria that should not have any antibiotic resistance, and they should not have the ability to make blue colonies. You will also have a control plate, which we'll talk about in the expected results, which will be the LB plate, meaning it's only got nutrient-rich media and it does not have anything bad in it. It does not have any XGAL to produce any colors either. Okay? Make sure you're comfortable with control groups. Now, whenever you do any experiment, it's very important to know your expected results, because if you do not know exactly what you're expecting, then how can you tell once you get results whether they're right or wrong? How can you troubleshoot or analyze anything? Okay, so when we talk about the expected results, we'll break it into two groups. You have your expected results for the control, again, remembering P blue is your control samples, so control group. Okay, and with that, after we'll go through the results, the expected results of when the plasmid is present, which we call the plus P blue sample. So when we do this experiment, we have 3 plates for our control group. We have the LB plate, we have the LB amp plate, so it has ampicillin, and we have the LB amp XGAL plate. Okay, and in each one you have to ask yourself, do I expect growth or no growth? And if I expect growth, do I expect it to be the normal whitish beige growth, or do I expect it to be blue? Okay, so first of all, we have our LB plate. This is nutrient rich with nothing bad. Okay, that means you expect any and all bacteria, whether it has the plasmid or not, to show growth. Okay, you expect growth, and you expect it to be normal, regular old, well, I'll call it normal beige growth, because there's no selectable markers to change the color. So even though there is no plasmid, because remember, we're dealing with the control group, so remind yourself up here, no plasmid in any of these samples, you expect growth on that first plate because of the fact that it's just nutrient-rich media. Now, LB amp, that means it has ampicillin, so now antibiotic present. What happens to these bacterial cells that have no plasmid, no beta lactamase? They die, okay? No growth. Cells should be killed, okay? Antibiotic present kills cells, okay? Because no plasmid to protect them. Okay, that then brings us to the last one, and this last plate has ampicillin and XGAL, but the XGAL doesn't matter in this, because as soon as you see ampicillin and you see minus P blue, you once again know that there is no growth, okay, because amp kills them. If you ever do an experiment where your minus P blue plates that have ampicillin suddenly show growth, that tells you a few different things, okay? When you're troubleshooting, ask yourself, what might be wrong with the bacteria? What might be wrong with the plate, okay? What would lead to growth in these presents? Now, the way to troubleshoot this kind of thing is you can think of each step in the protocol and where things can go wrong. Also, you can ask yourself, well, if I have ampicillin on a plate and I have no plasmid present according to my labels, what might cause growth on an ampicillin plate? Well, either the bacteria somehow got the plasmid, meaning maybe you swapped samples, maybe you wrote the wrong label, maybe you cross-contaminated, meaning you took a loop or a pipette from one sample and put it into your other sample, meaning you got accidentally tracked some plasmid in. Or maybe there's something with the plate. Maybe the plate was mislabeled and it was written that there's supposed to be ampicillin in it, but it was not. Maybe the ampicillin got degraded or destroyed, because the way that we prepare these plates is that we add the ampicillin while the agar is still liquid, meaning it's hot, freshly sterilized from an autoclave, so it's really hot. If you don't let it cool enough, then it's too hot that it denatures the antibiotic. If you let it cool too much, it starts clumping like jello because it wants to solidify. So it's a tricky thing to make these plates. So sometimes there could be a problem with the ampicillin in the plate. But ultimately, you have to think about what were you expecting and what could have gone wrong. This is important because I will ask you to do some troubleshooting in the results analysis. Okay, so make sure you pay close attention to the various things that can go wrong. Also think of every step in the procedure and where something can go wrong, okay? So now the rest of the results to analyze are your plus P blue samples. And for the plus P blue, I want to point out, you always want to circle it and remind yourself that these had plasmid added. So had plasmid added. So now hopefully they took in the plasmid, they replicated, they made the proteins, the beta-lactamase, the beta-galactosidase, and now you're testing to see whether that happened. Now you use the same 3 types of plates that you use in the control. You use LB, you use LB amp, and you use LB amp X gal. Okay, and now with this, you have the first plate. You are expecting any and all growth on that first plate because that's your control plate to see whether or not the bacteria were truly viable. Viable meaning, did they survive everything that you did to them? Okay, were they good, healthy cells to begin with? That way you know that if you don't get growth on other plates, on the selective plates, was it the fault of the cells, or was it that they did not take in the plasmid? Okay, so the LB plate is an important control plate, and you always hope or expect there to be normal growth. And again, normal growth is beige growth. Okay, and I'm not going to write underneath it why. You should already have, notes that is nutrient-rich media. It does not have anything bad in it, so everything should grow. What I will make note is the LB plate is to test if starting bacteria is viable, and that means healthy. Okay? Then you have the LB amp plate. So now there is antibiotic present. Okay, so you could always remind yourself, antibiotic here. Okay, and there's antibiotic in the last one as well. Now, the normal starting bacteria do not have antibiotic resistance, so they normally get killed on those plates. If, however, the bacteria took in plasmid, because this is the plus P blue sample, then what should happen on the LB amp plate? You should see growth, okay? Because P blue gives beta lactamase, okay? Inactivates AMP. So the bacteria can survive. Now, the next question I ask you, are you expecting that growth to be blue? What do you notice about this plate? What doesn't it have? Yes, you have the P blue plasmid, but what is not on the plate? There's no XGAL listed. So instead, you expect to see normal beige growth. And that is because no XGAL in plate. Okay? So you do not expect any color to the growth. So no XGAL in plate, hence why you expect beige growth. Okay, because the plasmid and the bacteria do contain at this point beta-galactosidase, but there's no XGAL for it to cut in half, so there's no blue produced. Okay, then you have the last plate, which is most important because it has ampicillin and XGAL. So yes, with the plasmid you expect growth because the P blue allows it to survive antibiotic for the same reasons that we wrote in the second one on this page, okay? But in addition to that, you now expect blue growth because XGAL is present. So survives antibiotic and beta-galactosidase cleaves XGAL. So you see blue growth. Okay, so the only time you should see blue growth on any plate is when P blue is present and XGAL is present. So for instance, if you were troubleshooting and let's say you had a plate that was labeled as LB or LB amp and you suddenly saw blue colonies, what would you say? Well, plasmid is present, and what else is present? XGAL must be present. That's the only time you'll see blue colonies. So in troubleshooting, you might say, well, they might have mislabeled these plates, swapped plates, because that plate clearly has XGAL. The bacteria has P blue, the plate has XGAL. I don't care what you wrote on it, you're wrong in whatever you wrote, okay? So in terms of the label. Okay, so things get mixed up, things get swapped. Sometimes, you know, people make plates and don't label them right. And so you see those kinds of results. So I want you to be very comfortable with troubleshooting. So always ask yourself, what causes the kind of growth I'm seeing? And what does that tell me about the labels that existed, about the samples that were used, about the steps that were taken? Okay, so the ultimate goal in the transformation experiment is to get that blue growth on that last plate, the LB amp XGAL plate. If you are uncertain of any of the expected results we talked about, simply contact me in the Remind app because the expected results and being able to analyze results is critical to understanding a transformation. And on the quizzes, on the post-lab, on the final, I can give you any plate. I could say, you know, there's an LB amp plate with a minus P blue sample, and I could describe a type of growth on there, and you should be able to tell me, is that the expected growth, or did something go wrong? And if something went wrong, what might it be? Would it be that there's XGAL there? Would it be that maybe there's P blue, no P blue? You know, you have to be able to analyze these things because those are the types of questions you get asked. If you want more practice beyond the post lab, simply contact me in the Remind app, and also never hesitate to contact me as you are doing the post lab. I will help you with those questions because it is not an exam. The post lab is part of the lesson, helping you to understand and better gain, comfort with the material. Okay. So now in your slides and in this presentation, I gave you a copy of the exact protocol that we usually use in the classroom in our genetics lab, so that you can take a look and kind of get used to seeing how the steps might actually look in terms of what you would do. Like I mentioned earlier, it always has the same key steps. For instance, first adding calcium chloride, incubating on ice, having heat shock, ice, the nutrient-rich media, which is the urea broth, okay, incubating for some time. Whenever you do these steps, even though the volumes, the amounts you're using, the times you're using may vary, the steps are always the same and the purpose is always the same. So when you go through a protocol, I want you to always ask yourself, why am I doing these steps? That's critical for being able to troubleshoot and also for being able to swap out certain reagents or adjust the protocol as necessary, because sometimes you don't have all of the supplies listed or whatnot. Okay, so for instance, when you do this room temperature incubation with urea broth, I would want you to know, well, why do we do that? That's the step so that the cells have time to replicate and, more importantly, transcribe and translate plasmid. So specifically, the whole point of that is so cells have time to make the protective proteins. Okay, so I know we already went through all of the steps, but I just wanted to give you an example on the protocol itself, looking at one of the actual steps, okay? And you can do that, practice doing that for each step. What's the point of that first calcium chloride, of that ice step, of that heat shock, of this ice step? Whenever you go through a protocol, practice that. We then have the remainder of the protocol. You notice that this, in the written out area, you'll see the heat shock that we talked about in the picture and all the other steps. You'll also notice the final incubation and the various plates, the selective plates that you will use. And I want to point out one thing on this protocol. Whenever you are plating samples, always make sure to use the clamshell technique. Okay, that's what we use not just in microbiology and genetics, but in all of our different scientific fields, because that minimizes contamination. What that means is you never completely open an agar plate cover. Okay, you don't ever separate the cover and leave it on the bench or anything, because those agar plates, for instance, the LB plate that you're going to streak, that's meant for growing bacteria. And there's bacteria all over the room, in the air, on your hands, on your gloves, coming out of your mouth. Now that you've lived through a pandemic, you understand this regarding bacteria, viruses, fungi, all of the microbes. Okay, so clamshell in labs is very important for what we call aseptic technique, meaning you don't want any extra bacteria getting on those plates. So you can write this as avoiding contamination. Okay. Sorry, my handwriting's a little sloppy today when I'm writing on the screen standing up. Okay, so take a second to look over the protocol so you kind of see what it would look like in the real life setting. Now, the last aspect of this virtual lab is that I am giving you various examples of actual results, okay? And I want you to analyze them. You're going to do that in the post lab. And like I said, I could give information in terms of a quiz where I describe a plate and you should be able to pick out the analysis. Now notice that the plates will always be labeled as to whether they're LB, LB amp, or LB amp and XGAL. They will also tell you the type of sample. So you notice all of these in the results set 1 are minus P blue, okay? So all 3 of these are minus P blue, which are your control set. Now when you see this beige smear of color, that beige smear compared to the see-through part of the plate, that tells you that this here and this here is growth, okay? Growth and growth. The clear parts are no growth. Now in pictures it's tough to tell, so I will tell you that this plate and this plate, they are no growth, okay? You do not see any, outlines. You notice on this plate how you can see those weird outlines. That shows a blob of growth. You don't see any circular colonies, which you'll see on the next slide. Okay, so I'll ask you to analyze these plates, and when you analyze any results, you want to be specific to each plate that you see. You want to be specific about what that plate contains, and you want to be specific as to was that what you were expecting or not, and what might have gone wrong. Okay, so analyzing includes all of those things. Make sure, for instance, when you are analyzing results set 1, you talk about the LB plate, the LB amp plate, and the LB amp XGAL. You talk about the fact that they are minus P blue. Now notice this LB plate also has half that has the plus P blue, okay, but that's for the separate analysis, so don't worry about that half on result set number 1. Focus on the minus P blue. Describe what you would have expected, what you got, and whether or not it's the same thing. You then have result set number 2. And both of these plates, I know they're a little blurry the way the students wrote on them and the way that I got the pictures, but notice both of these plates are plus P blue, okay? So both of them would be plus P blue and plus P blue. You already saw the plus P blue result on the first set, the one that I X'd out for set 1, that's actually part of set 2. Okay, so if you're asked about the LB plate for plus P blue, it is in result set 1, even though I'll talk about it as if it's result set 2. I want you to notice, it's a little blurry, but this plate on the left, this is the LB amp plate, whereas this plate on the right, this is amp and XGAL. And one of my former students, he did beautiful work in terms of lab reports and everything, and so he made this lovely figure where they even zoom in so you can actually get a better, closer look at the colonies, and you can see the difference between what this growth on the left looks like versus this growth on the right, and what you are seeing scattered throughout that growth, okay? And again, always ask yourself, what sample is it? What plate is it? What was I expecting? And what could have led to these results? Did they work, or might something have gone wrong? And that kind of troubleshooting is critical for the final result set that I will have you analyze. Again, these are all real, plates, real results. And notice each one you should be able to clearly read in the PDF slides or in this video here. You have the plate type and the sample on them. So this first one, LB with plus P blue sample plated on it. The second one is LB amp with plus P blue plated on it. And the third one is the amp and XGAL plate with plus P blue. all of the bottom three are labeled with minus P blue, okay, so no plasmid, and they are LB, then LB amp, and amp xgal, okay? I will point out again, because it's tough to tell in pictures, this is what growth looks like, this is what growth looks like, this is what growth looks like, and those little spots that you can tell what their color is, hopefully, if you zoom in on your PDF slide, okay, those are all growth. The two over here, okay, and I'll even change the color so that you see the ones that I'm putting in green here, okay, that is just condensation. There is no, sorry, the green does not come out right on here. There is no growth here, and there is no growth here. And so when you analyze this set of plates, I want you to analyze every single plate. Ask yourself, is it what you were expecting? If it's not, what could have gone wrong? What leads to that result normally? What does that tell you about the experiment? Okay, be as specific as possible. Make sure to mention things such as beta-lactamase, beta-galactosidase, ampicillin, xgal, plasmid, no plasmid, all of that stuff. The last thing I want to mention before letting you go in this video is when you look at each of these plates, you see that plastic outer rim. That is the cover of the plate, so they're currently flipped upside down in this picture. When you streak the plate, again, that cover, you always clamshell it, meaning if you have a plate, okay, and you're opening the cover, you only open the cover at like a 45-degree angle, okay, so that would be the cover, this would be the bottom of the plate. You never completely separate the cover from the plate, okay, so clamshelling is very important. And that is it for today. Like I mentioned, transformation is critical because it not only comes up in genetics lab and lecture, but it also comes up if you take microbiology, even some biochem labs, cell bio, the intro bios, and in grad school you do it as well. And no matter which field you go into, you end up relying on transformation. So please, please, please make sure you understand it very well. As always, contact me with any questions when you're analyzing the post labs or when you are studying the slides. Okay? Good luck and have a wonderful day.